Bismillahirrahmanirrahim, and welcome back to this next video. And this is the second video in the series of lectures on the immune system. In the previous video, I've told you that the immune system is a complex network of cell and proteins that defend the body against the infection. And the immune system comes in two flavors. One is known as the innate immune system, and the second one that is known as the adaptive immune system. So first we are going to talk about these first videos are about the innate immune system. In the previous video, I've told you that the innate, it means the inborn or natural immunity, and it is a non-specific type of the immunity which come into play immediately or within hours of an antigen appearance in the body. Now the immune system can be this innate immune system. It usually includes the physical barriers, the chemicals in the body, and it includes the immune cells. And in the previous video I had a detailed discussion on these physical barriers, and I'll share the link in the description. So in the physical barriers, I've told you that the skin and the other epithelial surfaces, they are actually working as a physical barrier, thereby separating the inside and outside of the body. And we talked about the junctions, the tied junctions at the adherence junctions, and we also talked about the interior epithelial surfaces which are actually having these mucins which physically help prevent the pathogens from adhering to the epithelium cells. And then in the last we talked about the defensin proteins in detail, and these defensin proteins, they are actually active against a broad spectrum of the microorganisms or the pathogens. Now in this particular video I want to focus on the human cells recognition, how the human cells, they actually recognize the conserved features of the pathogens. Now when you talk about the immune system, there should be something specific to the microorganisms or the pathogens that the human immune system can recognize and particularly neutralize only the pathogen without harming the host cells. So when you talk about the microorganisms, they sometimes do breach the epithelial barricades, and when they breach the epithelial barricades, then they are going to cause a disease in the human body, and that will be having some in the that particular individual will be having symptoms of that particular disease. Now when the microorganism, they breach the epithelial barricade, it is the duty of the innate and the adaptive immune system to recognize and destroy this pathogen without harming the host cells. So we are only focusing on the innate immune system in these particular videos, and in the following videos we'll be talking about the adaptive one. So when you talk about the innate or the adaptive immune system, they should distinguish the self from the non-self, because if the immune system cannot differentiate between the self, that means the cells of the host, from the non-self, that means that the pathogenic cells, that means that the immune system, while neutralizing the pathogen, can harm the host cells. So the immune system is good enough to distinguish the self from the non-self. Now when you talk about the pathogens, there are particular types of molecules that are common to pathogens, and they are not found in the host cells. What this means is that these particular molecules that can be recognized by the immune system and that can be used to neutralize the particular pathogen. So is these particular molecules, they are not present in the host cells, that means that any reaction that the immune system generates, that will not be affecting the host cells. Now these particular type of the molecules, they are called is the pathogen associated molecules, and the name is very clear. These are the molecules which are only associated with the pathogens. So the activity of the immune system, that will be very specific. Now these pathogen associated molecules, they are also known as the pathogen associated immunostimulants, because these molecules are actually stimulating the immune system. So they are also known as the pathogen associated immunostimulants. For short, I will call them as the PAIS, for the pathogen associated immunostimulants. Now, sometime in the literature, you will also found that these pathogen-associated molecules or these pathogen-associated immunostimulants, they are also known as the microbial-associated molecular patterns. So don't get confused in these terms, because all of these three terms, they actually refers to one in the same things. The particular molecules on the pathogens, they are actually stimulating the immune system of the host. Now, when you talk about these pathogen-associated immunostimulants, what they do is that they are actually responsible for stimulation of the inflammatory responses by the neutrophils, and they are also responsible for stimulating the phagocytosis by the macrophages. We will discuss both of them in detail. Now, both of these responses, whether that is the inflammatory response or that is the phagocytic response, that occur very quickly even if the host is never exposed to the pathogens, because these phagocytosis and the inflammatory responses, they are part of the innate immune system. That means they can recognize each and every pathogen even if the host is never exposed to that particular pathogen, because the innate immune system is not a specific immune system. Now let us talk about some of the important types of the pathogen-associated molecules. One of the important type of the pathogen-associated immunostimulants that is known as there are differences in translation initiation. Now, when a pathogen, say for example, if a bacteria, it enters into the host cells, so one of the phenomena by which the immune system can recognize them is that there are differences in the translation initiation of the prokaryote and the eukaryotes. When we talk about the translation initiation in the prokaryotes, the first amino acid in the prokaryote is actually a formylated methionine. When you talk about the first amino acid in the host, like the human beings, they are actually using a regular methionine in their translation initiation process. This means that the transcripts, or the you can say the polypeptide that is made, so the first amino acid at the end terminus of that particular peptide in the prokaryotes, that will be having a formylated methionine, and when you talk about the host cells, they will be having a regular methionine. So this is one of the important difference in the translation initiation of the prokaryotes and the eukaryotes. So any peptide that is having a formylated methionine at its end terminus, because the end terminus is the start position of the polypeptide. So if the end terminus is having the first amino acid as formylated methionine, this means that peptide will be of the bacterial origin, and these formylated methionine, they are of the bacterial origin, so they are actually working as a chemoattracting for the neutrophils, because the host cell that is having a regular methionine, so those peptides, they are not recognized by the immune system, but is the peptides of the prokaryotes, they are having their first amino acid as formylated methionine. So these formylated methionine will be acting as a chemoattracting for the neutrophils. The neutrophil is going to migrate towards the source of the peptide, and the source of the peptide, of course, will be the pathogenic bacteria that have entered the body, and when the neutrophils recognize them, they are going to neutralize the bacteria. So this is the first type of the pathogen-associated immunostimulant, that there are differences in the translation initiation of the prokaryote and the eukaryote, and the immune system can actually exploit this difference for neutralizing the pathogens. The other types of the pathogen-associated immunostimulants is that there are different molecules on the surface of the microorganisms which are not present in the host cells. So when you talk about the surfaces of the microorganism, the bacteria, whether that is the gram-positive or the gram-negative bacteria, they have got a peptidoglycan cell wall. So as the host cell do not have a peptidoglycan cell wall, the immune system can actually exploit this peptidoglycan cell wall to recognize the bacteria and thereby neutralize them. Of course, there are differences between the peptidoglycan cell wall of the gram-positive and the gram-negative bacteria, but we are not going into that. So the peptidoglycan cell wall, this is another important pathogen associated immunostimulant which can be recognized by the innate immune system. The flagella of the bacteria is another important molecule on the surface of the microorganism which the innate immune system can recognize. When you talk specifically about the gram-negative bacteria, they have got a special molecule on their surface which is known as the lipopolysaccharide, and the immune system can actually recognize this LPS. How the immune system recognize them, we will discuss them in detail in the next video. When you talk about the specific or molecules on the surface of the gram-positive bacteria, they have got a teichoic acid on their surface, and that can actually work as pathogen associated immunostimulant for the innate immune system. When you talk about the cell wall of the fungi, they have got special molecules like the zymosin, the glucan, and the chitin. The important thing about all of these molecules is that they are absent from the host, so any immune system generated against them will not be affecting the host cell. They could be specific against the microorganisms. When you talk about the parasites, they also have got a unique membrane components. Like for example, if you talk about the plasmodium, they have got a special molecule on their surface which is known as the glycosyl phosphatidyl inositol, and the innate immune system can actually recognize this as a molecule to neutralize it. Another important thing in the bacteria, there are short sequences in the bacterial DNA which are known as the CpG motif. This is the cytosine, this is the guanine, and they are connected to each other by this phosphate group. So this is known as the CpG motif. Now, the special thing about this CpG motif that it contains unmethylated dinucleotides. These two, these C and this G, of course they are the dinucleotide. So this CpG motif actually contains these unmethylated dinucleotide, and they are flanked by two purine residues at its 5' end and two pyrimidine residues at its 3' end. So say for example, if this is the 5' end, so you will be having two purine residues followed by the CpG motif, and that will be followed by two pyrimidine residues at its 3' region. Now, the important thing is that this CpG motif, the unmethylated dinucleotide, it is actually 20 times less common in the vertebrate DNA. So this CpG motif in the bacterial DNA will be working as PIAS, like the pathogen, the PAIS, the pathogen associated immunostimulant, and the immune system will be using this CpG motif to recognize the bacterial pathogens. Now this CpG motif is actually responsible for activation of the macrophages. It can also stimulate the inflammatory responses by the neutrophils, for example, and they can also increase the antibody production by the B cells, and of course that is part of the adaptive immune systems. Now most of these pathogen associated immunostimulants, they occur on pathogen surface in repeating patterns. So when they occur in repeating patterns on the surface of the pathogens, they are actually easily recognized by the innate immune system. Now, when these pathogen associated immunostimulants, if they are present on the surface of these pathogens, what the host do against them. So these pathogen associated immunostimulants are actually recognized by several types of dedicated receptors in the host, and these dedicated receptors which recognize the pathogen associated immunostimulants are known as the pattern recognition receptors, and we will discuss about the pattern recognition receptors in the next video. So if you like the video, please subscribe to my channel, hit the like button and share it with your friends.